Hey guys, this is Iqbal and today we'll be going to study about the process of transcription and translation. Transcription is when DNA molecule is going to make the copy of RNA, and translation is when RNA is going to make the copy of a protein or a polypeptide chain. So when we try to have a look into the structure of a DNA and the main structure of DNA, we have studied in a previous video. If you want to know about the structure of a DNA, please refer the previous video, which is the structure of a DNA. And on DNA, these nucleotides are arranged in a specific manner, and especially a region on the DNA which is coding for one protein is called as a gene. And these nucleotides, they are arranged in a group of 3, which is called as a triplet, and one triplet is a code for one amino acid. So in this way, many triplets will be joining together to make one gene, which is a code for one specific protein or one polypeptide chain. So then we can say that especially the sequence of nucleotides on DNA is called code, and the sequence of nucleotides on messenger RNA or RNA is called as codon, and the sequence of nucleotides on transfer RNA is called as anticodon. Let's have a look into the process of DNA transcription. And DNA transcription means when a DNA is going to make the copy of messenger RNA. And here in front of us, there is one cell, and this is its cell membrane in the cytoplasm, and then the nucleus and nuclear membrane. Inside the nucleus, there is a genetic material. Mainly in genetic material or in the nucleus is a chromosome. There are different number of chromosomes with the different species, and if we look into the human, there are total 46 chromosomes in human being. And a chromosome is actually the combination of a protein which is called histone and DNA, and DNA is wrapped around the histone proteins in such a way, making the nucleosomes. So when histone protein is surrounded by a double strand DNA molecule, this is called a nucleosome. Many nucleosomes will be combined together and will be condensed and packed in making structure of a chromosome. So let's go and look at that, how a DNA undergoes a process of DNA transcription, mean making the copy of an RNA. So to know more about the structure of DNA, please refer my previous video, which is about this. The name of the video is structure of DNA and RNA. And when DNA is going to start the process of DNA transcription, mean making the copy of an RNA, so the first thing which arises on DNA is an enzyme called RNA polymerase II. This enzyme come and attach to the DNA, unwind it, and then start adding RNA nucleotides. So the difference between the nucleotides of DNA and RNA is the presence of uracil instead of thymine, and also, as we know in the previous video, that 5-carbon sugar at carbon number 2 have oxygen for RNA but not for DNA molecule. So in initiation stage, then addition of these nucleotides will be continuously going on, and elongation increasing the size of the RNA molecule, and finally RNA will reach to one specific sequence of nucleotides on DNA where the process will be terminated, and a piece of RNA will come out from or will be broken down from the DNA and making one pre-messenger RNA. This pre-messenger RNA, as it come out from or removed from the DNA molecule, so it will be processed. Mean one on its 5' end GPP cap will be added on its 3' end. A poly A tail, mean many molecules of adenine nitrogenous base will be added. And main function of this tail and cap is to protect the pre-messenger RNA from degradation. But it will not stop here. This pre-messenger RNA will go to the further process also. And if we look in deep into the structure of this pre-messenger RNA, it consists of introns and exons. So in this case, a process called splicing will take place, and these introns will be removed and exons will stay together, and a mature messenger RNA will be formed. So this is the total process of transcription, where a final messenger RNA is formed. This messenger RNA is now ready to leave the nucleoplasm, and nucleus, and to enter into the cytoplasm to go for the process of translation. As messenger RNA enter into the cytoplasm, first thing, it will be welcomed by a ribosome. The smaller unit of ribosome come and attach to it, and then it will be followed by the larger unit of ribosomes. Ribosomes are actually also made from rRNA or ribosomal RNA. They are not proteins, they are made from the RNA by the same process of DNA transcription. And splicing, adding of tail and cap, everything will take place with the formation of ribosomal RNA also. So these ribosomes, when smaller unit of ribosome attach and the larger unit, so we see on the larger unit of ribosome there are 3 places like A site, P and E. A site is where this first transfer RNA come and attach to it and give amino acid. Then it moves to the P site and finally to the E site to exit. In cytoplasm, there is another type of RNA called tRNA or transfer RNA. This transfer RNA come and attach to the active site of an enzyme called amino acyl transfer RNA synthetase, and then an amino acid which come from our digestive system into the cytoplasm of the cell. One of the amino acid come and attach to the 3' end of transfer RNA with the help of an enzyme called amino acyl transfer RNA synthetase. Then this transfer RNA moves to the A site of the larger unit of ribosome, and its anticodon is the complementary with the codon of the messenger RNA, and the start codon on messenger RNA is AUG. Now messenger RNA will move further, taking this transfer RNA to the P site, and now A site is free. This now another transfer RNA come take one of the amino acid with the help of the enzyme and come attached to the A site of the ribosome, where now this another thing will happen, that the transfer RNA which is present on P site will give its amino acid to the transfer RNA on the A site and move to the E site or the exit site. There will form a bond or a peptide or a covalent bond will be formed between the amino acid of which is left from the first transfer RNA and the transfer RNA which is on the A site, And now again this messenger RNA will move further, taking this transfer RNA to the P site. When it reaches to the P site, then E site transfer RNA will leave, and on the other side another transfer RNA will come and try to get an other amino acid from with the help of the enzyme and attach to it and then take to the A site of the ribosome. And when it take to this to the A site of the ribosome, again this anticodon and codon will be having a complement. They will be the complementary of each other. So now this again the transfer RNA which is present on P site is going to leave and going to move to the E site, and the transfer RNA which is present on A site will, their amino acids will combine and it will move to the P site, carrying all that peptide chain with it. Now transfer RNA which is present on E site will leave it, and a transfer RNA which have taken an amino acid with the help of an enzyme will come on the A site. And when it reaches to the A site, then again messenger RNA will move a little bit further, and the transfer RNA which is present on P site will move to the E, and this transfer RNA which is present on A site will move to the P site, having a polypeptide chain with this. Now here a final a termination codon will come on the messenger RNA, which will terminate the process of this translation, addition of these amino acids on it, and then the transfer RNA which is present on exit site will leave, and also the transfer RNA which is present on P site, that also will go to the E and finally will leave it. And now we at the end we have finally a chain of amino acid or a polypeptide chain which is formed by a process of translation is there with us. This protein or a polypeptide chain now is ready to move from the cytoplasm. First, this will go to the endoplasmic reticulum and Golgi apparatus, where it will be processed and packed and then transported to the different places in the body where it is needed.